We can see it. Go ahead, Gerardo.
Okay, thank you.
Oh, you know what? Yeah, go ahead and put it as presentation. That's great. Now we can see it.
Okay, great. Thank you. And thank you all for attending this workshop. My name is Gerardo, and I'll talk to you briefly about the project we've been working on by the direction of Dr. Samuel Perez. Okay, so as many of you know, nowadays nutritional tendencies are leading us to some bad nutrition because of socioeconomical problems or just simply by bad habits. And this bad nutrition can also leads us to illness that are not limited to obesity or malnutrition, but can also lead to some heart conditions, some blood diseases, tissue diseases. And well, to prevent that, the food industry are adding vitamins, minerals, and some other molecules that can help our bodies to stay healthy. And this is the case of the coenzyme Q10. Well, the coenzyme Q10 is a molecule present in the mitochondrial membranes, and it's active in the electronic transfer chain. And it can also be present in some oxidation reactions, like an antioxidant. So coenzyme Q is present in almost any living thing, but we as humans only have the Q10. The Q10 stands for the 10 units isoprenyl chain, and also made by a benzoquinone ring, which is the one that participates on the oxidation reduction reactions. So, well, this molecule is relevant because it's some studies have shown that it can help with some energy deficiency in the cells, because, well, the electron transport chain is one of the main sources of energy for any living organism. So when you take a daily intake of the coenzyme Q10, it can help you to prevent or to reduce some of the diseases, even Alzheimer, some heart conditions, and some other illnesses. So, well, even though we can find the coenzyme Q molecules in any living thing, like I said, we can have some different ways to get it. It can be through chemical synthesis. It can be through the tissue production, animal tissue production, sorry, and also by microbiological production. And well, some studies have reported that the microbiological production is the most feasible and the easier way to get it. So that's why we chose this yeast called Sporidiobolus johnsonii, because it's the one that produces exclusively coenzyme Q10, because it has been under strain select, and they reach a strain that produces exclusively the coenzyme Q10, which is the one that we have on our bodies, and relatively short times. And well, some facts that can help us to work with this yeast is that it has a distinctive pink color. And well, how are we going to get it from the yeast cells? We're going to make an ultrasonic assisted extraction, and this is because the ultrasound cavitation can help us a lot with the extraction from inside the cell. Because as you can remember, well, the coenzyme Q10 is mainly inside the membrane of the mitochondria. So we have to penetrate the cellular membrane and also the mitochondrial membrane. So how does the ultrasound cavitation helps us? Well, it works under a compression refraction cycles that produces bubbles. And well, when the bubbles becomes unstable, they collapse and they implode. And this can cause different kind of high energy phenomena in the medium, are like shear forces that can help to lysis the membrane, and also some sun operation that can help to the solvent to diffuse, Also some micro mixing that can help to the solvent to interact with the molecules faster. So our hypothesis is that ultrasonic assisted extraction is an effective alternative to obtain the coenzyme Q10 from the Sporidiobolus johnsonii production. And our main objective is obviously to provide an ultrasonic assisted extraction to obtain the coenzyme Q10 from the yeast. So some of our specific objectives is ultrasonic assisted extraction operation variables, the ones that are the most critical in the extraction. After that, we're going to do a Taguchi design of experiments to see if all these variables are relevant or significant to the extraction. Then we're planning to do a surface response optimization. And after that, we're going to run a simulation of the conditions that we come up to. We're getting to the methodology section, and we're starting the biomass production. To make the biomass that we're using to extract the coenzyme Q10, we take it from a cryopreservation tube, and we add it to a Petri dish with YM agar under the conditions of 30 Celsius degrees for 48 hours. After that, we put some of these cells we produced on 10% of the total volume we're going to do on the fermentation. It could be on YM medium or agro-industrial waste medium under the condition of 30 Celsius degrees for 18 hours. Then we add the inoculum to the total volume of the same medium I talked to you about earlier, and it goes under the condition of 30 Celsius degrees and 48 hours. After that, after the fermentations are done, we collect the biomass by centrifugation at 10,000 RPM, and we collect the biomass pellet. We made our reactor type fermentation. Is done on a solvent flask, and the conditions we used was 2 liters of agro-industrial waste medium, external aeration of 0.97 ppm of air. It went under bubble agitation, and the conditions were 38 Celsius degrees and 48 hours. Then we reached the ultrasonic assisted extraction. The equipment we used was an ultrasonic processor, a water pump, and a stainless steel extraction chamber. It has like a jacketed reactor shape. And the water pump was used to maintain the temperature on the extraction chamber. Well, after that, we dried our biomass. Our biomass was homogenized. Then it was put onto Petri dishes in a forced convection oven under the condition of 60 Celsius degrees for 6 hours. After that, we grind it on our mortar and pestle. And we ran a dried biomass extraction, but a conventional extraction, not an ultrasonic one. And we added 1 gram of the dried biomass on 50 milliliters of solvents. It was a 1:1 xn ethanol ratio at 100 RPM agitation for 1 hour. Then we centrifugated the extraction at 10,000 RPM, and we collected the supernatant. After that, we put it on a rotary evaporator with vacuum, 60 Celsius degrees, 200 RPMs, and then we resuspended the solids with 5 milliliters of ethanol. And the spectrophotometric quantification we made was with an UV visible spectrophotometer by Perkin-Elmer, and the conditions were a wavelength of 275 nanometers. We used a blank of ethyl alcohol and external standard of coenzyme Q10. We ran also a fractional factorial design of experiments to check on the effects of 3 factors, which were ultrasound treatment, time, ultrasound amplitude, and hexane ethanol composition on the solvents. We use a two-level factorial design, and on steady, we put a half of a gram of biomass and 15 milliliters of total volume. Well, the results we have come up with, well, I didn't put this exactly on the methodology, but it's something that we've been working on because when we first started, we started producing the biomass on Erlenmeyer flasks, and they were like 500 milliliters or 250 milliliters. So here we can appreciate different production methodologies, which were on Erlenmeyer flask, Falcon tubes, on Durant flask, and also the reactor type arrangement I talked to you about earlier. And well, what we can see here is that on the first graph on the left side, the max biomass yield was in the reactor type arrangement. And on the right side, even though the Erlenmeyer flask was having a better yield of biomass production, because of the volume we were making at each fermentation, it was very low amount of grams of production. And to make the design of experiments, we need a lot of amount to run it. And this is why we came up with the reactor type arrangement. And what else can I say about this? Well, that was it. Mainly, it was a change in the production methodologies, and we almost reached the bioreactor methodology. Okay, then after we ran the ultrasonic assisted extraction design of experiments, we made the statistical analysis, and we ran an analysis of variance on the model and also with the factors. And as we can appreciate here, as the model and the factors were significant to the model, and what can we get from this? That the model we're using, it can predict on the extractions. We use mainly 4 treatments, and there you can see the conditions we used. And well, we ran after, on the results, we ran a 2-key mean comparison. So what can we appreciate here? On the treatment number 3, it has the highest production, well, the highest extraction of coenzyme Q10, and is significantly different from the other ones, as the treatment number 2 and 4 are not significantly different, neither the treatment 4 and 1. But what information can we get from here? That treatment number 3 was the best one. Why is this? Because as you can appreciate, on treatment number 3, we had low levels on the ultrasonic treatment and high level on the solvent composition. This means that even though ultrasound is very helpful on these extractions, it's also very aggressive on the molecules. And the more time it interacts, or you have applying this energy to the system, is not selective. So it's going to be destroying all kind of molecules, even the coenzyme Q10. And it goes the same on the ultrasound amplitude. This is the power of the probe. So the more energy you put into it, is gonna start tearing apart all stuff inside the system. So this is why treatment number 3, which were the low levels on the ultrasound and the high level on the hexane ethanol composition, was the best one. And also because the molecule, the coenzyme Q10, is very lipophilic. So the hexane has an organic solvent, so it has a better affinity to it. And well, we can appreciate better here in the interval plot of treatments. It shows the amount of coenzyme Q10 extraction. And well, and you can see with the standard deviation, that is why the treatment number 3 was significantly different from the other ones. We also ran a tiny extraction, well, not tiny, but with only a few experiments of the dry biomass extraction. And well, sorry, I didn't mention it, but this design of experiment was done with wet basis biomass. So the first one, we ran it with wet basis biomass, and then we're planning to run the other one with dry basis biomass. So we ran a short experiment to see how much was the coenzyme concentration when you ran on a dry biomass extraction. And we get the results of around 15 milligrams of coenzyme Q10 with a standard deviation of 2.3. Our pending tasks are to do the HPLC methodology of quantification. We did one, but it was showing 2 different peaks, even though we were using a standard. What we're assuming is that the coenzyme Q10 is having different state of oxidation. So that is why it's showing different peaks. So we're planning on doing an oxidation of the whole thing. So it can be under one oxidation state and it can show just one peak. So we can quantify everything on one single peak. Then we're about to do the Taguchi design of experiments with the dry biomass. After that, we're planning to optimize the extraction and to simulate the process. This is the bibliography we've been using. And I'd like to thank you for listening to my presentation. If you have any questions, please let me know.
Thank you very much, Gerardo. Very nice presentation related to the extraction of coenzyme Q10. So Gerardo is open for questions, comments. Anyone would like to participate? We have 2 minutes time.
Hi, Gerardo. I have a question.
Okay, go ahead.
Patty, we cannot hear you. You're muted.
What kind of agro-industrial waste are you using?
We're using I forgot the name in English, sorry. Let me check it real quick, the name in English. I'm sorry, I got nervous. I forgot the name in English.
No problem. Do you know it in Spanish?
It's melaza. I forgot how is it called in English. It's almost the same.
Yeah, it's the same. Molasses.
Oh, molasses. Molasses, yeah. Sorry, I forgot. Molasses and also corn syrup.
Okay, and other question.
Patty, we're having problems listening to hearing you. Can you repeat that question, please, or write it in the chat? That's better.
Okay, I will write in the chat, okay?
Thank you. In the meantime, last question, Govinda is asking, Gerardo, did you experience any uneven surface at the base of the ultrasound probe after the continuous use?
No, I'm basing this on the results we had, and it didn't show this kind of effect. So, but I'm mainly based on the results we had.
Okay, and Patty, this will be the last question. Do you know how many genes are involved in the Q10 production?
How many what, sorry?
Genes. Genes.
Okay, no, I'm not aware of that. I'm mainly focused on the extraction right now. Our plan is to, well, by the end of a series of investigations, we're going to apply it to a food or as a supplement. But right now, I'm mainly focused on the extraction, so I haven't studied that, sorry.
No problem. Very nice presentation, Gerardo. There was a last question. We don't have any more time, to be fair to all presenters. So please join me giving an applause to Gerardo. Thank you very much.